Today's lecture is on cell cycle. So what is cell cycle? Cell cycle is a sequence of events which occurs during cell growth and cell division. This cell cycle consists of two important phases, the interphase and cell division. More than 70% of the period taken during cell cycle is completed by interphase and remaining period is by cell division. During cell cycle, all the preparation which are needed for the division of the cell to two daughter cell in case of mitosis, which occurs in the somatic cell, and one mother cell, gamete mother cell, myocyte, divide to form four daughter cell in the case of germline cell. So the preparation which needed to produce a daughter cell is occurs in the interphase. And now, what is interphase? The interphase is a cycle of event which helps in the preparation, once more, the preparation of all the necessary items like protein, enzymes, DNA duplication, the duplication of the cell organelles, the growth of the cell and the membrane occurs during this interphase. Now this interphase consists of the G1, S, G2 and G0 phase. So G0 phase is a quiescent stage, that is a suspended state. Suspended or the quiescent means once the cell division occurs, the cell divide, form two daughter cells are formed, they mature and they start functioning, and beyond which they never go again for division because they lost the capability of division, but they can repair if anyway they get damaged. So the cell undergo the G0 phase. Example of G0 cells are nerve cell, RBC, muscle cells and cardiac cell. So this cell undergo enters into quiescent or suspended stage. The G1 phase. In G1 phase, the gap bond phase, during this period, all the necessary preparation is done which helps to duplicate our DNA, the histone proteins and the centrioles. Here the cell organelles also get duplicated and the cell enlargement occurs due to the increase in the cell membrane. And thus the cell start producing all the necessary items which lead to the next stage, that is the synthesis phase. During this synthesis phase, the DNA get duplicated, the histone protein also get duplicated and centriole also get duplicated. Now the duplication of DNA requires many enzymes, many nucleotides and many cofactors which are produced during G1 phase. Histone protein is a DNA binding protein, is a positively charged and DNA is negatively charged. So the histone protein helps the DNA to wrap around itself so that the DNA get condensed into the form of chromosome, because in the interphase the DNA are in the thread-like structure that is called the chromatin fiber, which during mitosis prophase condense to form the chromosome. So the DNA which present in the one copy divide to form two copy attached with the centromere, and thus duplication of DNA occurs during S phase. Now in G1 phase all the cell organelles get duplicated, but since the DNA synthesis of mitochondria and chloroplast occurs just at the junction of the completion of S phase and start of the G2 phase, the duplication of mitochondria and chloroplast occurs at G2 phase. Okay, because the mitochondria and chloroplast contain DNA, the genome, the circular DNA, one molecules of circular DNA, and they are called extranuclear genome. So the cell enlargement occurs, all the necessary duplication of the cell organelles are made which are left in the G1 phase, and the cell ready for the division. Now, and as I told earlier, the interphase is the most active phase of cell cycle because it prepares the cell to divide. All the necessary compounds, suppose in the case of cell mitosis we need spindle fiber, the microtubules is a protein, microfilaments is a protein, all these are produced during the interphase. Now control of cell division is very important, because if the cell division is not controlled, regulated, there might be cancer and other genetic disorder. So there are two checkpoints, there are generally three checkpoints, one at G1 phase, one in G2 and one during the M phase. What is checkpoint? Checkpoint is a point, is a not any point or anything, but it is a regulation, is a protein like P53 is a protein which helps to check the integrity of DNA. Check the integrity of DNA. Suppose the DNA which comes from the cell will divide in the S phase, and if the DNA is somehow damaged, the damaged DNA will pass to the S phase, and this again pass to the G2 phase and then to the mitosis, and the daughter cell will contain the damaged DNA. This damaged DNA may cause cancer and many genetic disorder. So if a DNA has certain damage and mutation, checkpoint stops cell from further progress and give time to repair the DNA. And if the DNA get repaired, then the checkpoint allow the cell of further stage, that is the S phase, and thus the DNA will duplicate. If the DNA damage is large and could not able to repair itself, then programmed cell death occurs. That is called the apoptosis. So what is apoptosis? Apoptosis is a natural phenomena where when the cell have lots of DNA damage, and thus if it allowed to go for the next generation, it may lead to cancer, genetic disorder. So the P53, which is called the guardian of the cell, asks the cell to undergo apoptosis, and thus the cell is not allowed for cell division, and thus our body is saved from the abnormalities. So P53 protein produced from P53 gene and is considered the guardian of the cell. It regulates the cell cycle and cell growth. So the checkpoint 2, during checkpoint 2, which is present in the G2 phase. So in S phase, the duplication of DNA occurs, the DNA duplicate. So during replication, the DNA may get damaged. Suppose A is paired with T, and G with C. By mistake, if A is paired with C, so this is abnormalities. So all this will be checked in the checkpoint at G2 phase. And if during DNA duplication at S phase, there might be errors occurs which may cause genetic damage. So in G2 phase, the checkpoint 2 helps to monitor the DNA integrity. Now there are two important proteins, the cyclin and cyclin dependent kinase helps in the regulation of this checkpoint. There is another more checkpoint which occurs during the M phase and regulates the functioning of the cell division. So thus the cell cycle helps in proper regulation of cell division, because if there is any damage in the DNA, the cell will have the genetic disorder. If it allowed to go for the cell division, the daughter cell will contain the DNA damage. So the checkpoint helps to repair the DNA and then allow the DNA to progress to the next generation.